have looked up albinism on the internet.
Oh, and what did you find out?
Well, I found that the cause for dark skin is a pigment called melanin, and a lack of melanin causes people to look whiter. Hence, albinism is a condition when someone has less or no melanin.
That's true. Albinism is derived from the Latin word albus, which means white. People with such conditions have little or no melanin production. That's why their eyes look pale blue or pink. Some may also have red pupils and very pale skin and hair.
Wait a minute. Melanin is also present in the eye? I thought it's exclusive to skin.
Yes, it is found in the light-sensitive tissue at the back of our eyes and plays a role in normal vision.
I see. Does this mean albinism can also cause poor eyesight?
Yes, it not only leads to poor vision, but also causes rapid, jerky movement of the eyes. With such vision, people often have difficulty seeing under direct sunlight.
That sounds like a royal pain to deal with.
Indeed it is. Also, there is a different form of albinism known as ocular albinism, which affects only the eyes, but not the skin. And this condition is sex-linked.
Hang on. Sex-linked means the gene is linked with either X chromosome or Y chromosome. So albinism is genetic.
Definitely. It is a result of mutation in the gene.
I know about mutation. It is a sudden inheritable change in the base sequence of a DNA molecule.
Excellent. You see, mutations at several loci might be responsible for albinism. Also, since the mutated gene is autosomal recessive in general, only the individuals with homozygous recessive alleles show this condition.
Homozygous recessive, characterized by two lowercase letters for recessive alleles. Isn't it so?
Yes.
That means albinism must be a rare case, right?
Yes, indeed. 1 in 17,000 births might have it. However, the condition is relatively common in some populations such as the Hopi in Arizona and the Cunasan Bluffs Indians in Panama.
Got it. But teacher, is this only seen in humans? Because by the characteristic peculiarity of albinism, I think white rabbits are albino, and so are white mice.
You're correct. Since it is related to genes, we can observe albinism in different animal species too. It's seen in rabbits, kangaroos, and even tigers.
Oh, I've never seen a white tiger. It's so cool.
Yes, it is. To add on, albinism is a good example to explain the relationship between a gene, a protein, and phenotype.
I'm familiar with the role of genes in determining phenotypes, but how are the proteins related?
Let me explain. In albinism, a gene called TYR plays an important role. The TYR gene provides instruction for making an enzyme called Tyrosinase. It is an oxidase enzyme and has two copper atoms in its active site that bind with an oxygen molecule. Tyrosinase is a transmembrane protein and is found in the membrane of melanosomes. Melanosomes are large organelles that produce melanin and are found in specialized cells called the melanocytes. The enzyme tyrosinase is responsible for the melanin production from the amino acid tyrosin. Tyrosin in the presence of tyrosinase produces dopa, which then forms dopaquinone and finally melanin. But a mutation in the gene that codes for the enzyme tyrosinase results in either the absence of tyrosinase or the presence of inactive tyrosinase in the melanocytes. This inhibits melanin production.
Oh, I see. Then the two homozygous mutated genes give albino offspring, right?
That's correct.
So, this enzyme is found only in animals, including humans?
Not really. Tyrosinase can be found in both animals and plants. I'm sure you've seen how a slice of potato turns black when you expose it to the air.
Oh yeah, I've seen it in apples too, and they don't look good.
Yep. Apples and potatoes both have polyphenol compounds which bind with the oxidase enzyme tyrosinase to form dark-colored compounds as a result of enzymatic browning reaction.
Oh, I think I remember this. All these conversions happen in the presence of air or oxygen. This is an example of an enzymatic browning reaction.
Yes, that's right. Let me share some more examples that should help you understand the relation between genes, proteins, and phenotype better.
Okay, sure.
For that, you'll have to know 3 different conditions. They are sickle cell anemia, hemophilia, and Huntington's disease. For each of these phenotypic conditions, a specific gene and a specific protein are responsible. The HBB gene and hemoglobin are related to sickle cell anemia. The F8 gene and factor VIII are related to hemophilia, and the HTT gene and Huntington are related to Huntington's disease.
I know the relation between hemoglobin and sickle cell anemia, and also between factor VIII and hemophilia. But what's the role of those genes?
As you know, genes play a major role in protein formation. For example, the HBB gene codes for beta-globin, which is a component of hemoglobin. Does that ring a bell?
Oh, yes. I remember that hemoglobin is made of 4 polypeptide chains, each with one iron containing heme group in the center. Among those 4 polypeptide chains, 2 are called alpha chains and 2 beta chains. So this HBB gene contributes to beta chain formation.
That's correct. Therefore, a mutation in the HBB gene changes the amino acid sequence which forms the beta-globin in hemoglobin. As a result, distorted hemoglobin is formed.
Ah, yes. I remember that glutamic acid in the amino acid sequence forming hemoglobin is replaced by valine in sickle cell anemia. Actually, thanks for the refresher, teacher. I remember so many things now.
Great. So go ahead, it's your time to shine.
Sure. The normal amino acid sequence for the hemoglobin molecule is coded by the allele hemoglobin A. Similarly, the sickle cell amino acid sequence is coded by the allele hemoglobin S. This happens when the base sequence of CTC is replaced by CAC due to the substitution of nucleotides in a DNA molecule. Likewise, in the homozygous recessive condition, the 2 copies of hemoglobin S alleles lead to the genetic disorder called sickle cell anemia.
Bravo, you explained it pretty well.
Thanks, teacher. I have a question though.
What is it?
How is sickle-shaped hemoglobin different from normal hemoglobin? Doesn't it also participate in oxygen transport?
Good question. A very small difference in the amino acid sequence makes only a little difference to the hemoglobin molecule when it is combined with oxygen. But when it is not combined with oxygen, the unusual beta-globin polypeptides make the hemoglobin molecule much less soluble. So, the molecules tend to stick to each other, forming long fibers inside the red blood cells. As a result, the red blood cells are pulled out of shape into a half moon or sickle shape. When this happens, the distorted red blood cells become useless at transporting oxygen. They also get stuck in small capillaries, stopping any unaffected red blood cells from getting through.
Oh, I see.
A person with this unusual beta-globin may suffer from severe anemia, and it may be lethal due to lack of oxygen being transported to the cells.
Oh my God, that's really unfortunate. But teacher, what about alpha chains? If they are not regulated by the HBB gene, then which gene is responsible?
It's the HBA gene that is responsible for alpha chains.
I see. A in HBA is alpha, and B in HBB is beta, right?
Good way to remember. Now let's proceed to our next gene, protein, and the phenotypic condition.
You mean the F8 gene and factor VIII protein in relation to hemophilia?
Yup. Can you tell me what you know?
Sure. The F8 gene provides instructions for making a protein called factor VIII, which is responsible for blood clotting.
Yes, therefore, a mutation in the F8 gene leads to the production of an abnormal version of factor VIII, or it reduces the production of factor VIII protein.
And as a result, the blood clotting process cannot occur properly as a response to injury, and a person could die due to excessive bleeding, right?
No, not all hemophilic people die. There are levels of hemophilia depending on the type of mutation occurring on the gene.
Oh, I didn't know that.
Actually, you don't know a lot of things we're going to discuss right now, which is why this class is needed.
Of course.
Okay, let's get back on track. Some of the mutations only change single DNA base pairs in the gene, while others delete or insert multiple base pairs. The most common mutation that occurs in people with severe hemophilia is the rearrangement of genetic material called an inversion mutation.
Wow. The genes can invert. That's new.
Yes, this inversion mutation involves a large segment of gene which almost completely eliminates the activity of factor VIII, resulting in severe hemophilia, which can lead to the death of the person. Whereas other mutations do not eliminate but reduce the protein's activity, resulting in mild or moderate hemophilia.
The inversion is scary too. Also, I think hemophilia is a sex-linked trait and can only be expressed in a homozygous recessive condition.
Correct. Now let's move on to the next one.
It's some hunting disease, right?
Not hunting, it's Huntington's disease and got nothing to do with hunting. It is named after George Huntington, who was the physician who first described it. It is also called HD in short.
So teacher, what exactly is this disease?
It is a hereditary neurological disorder which results in involuntary movements and progressive mental deterioration. In this condition, the brain cells are lost and the ventricles of the brain become larger.
That's dangerous. But teacher, I thought we lose brain cells only when we are old?
Not really. The age of onset of this disorder is rather variable, but occurs most commonly in middle age. So, individuals may already have children before they know that they themselves have the condition.
That's bad, because it can potentially continue on for generations without people knowing.
Indeed.
So, what kind of mutation is seen in this condition?
Well, we can see the mutation as an unstable segment in a gene present on chromosome 4 that codes for the protein Huntington. In people who do not have HD, the segment is made up of a small number of repeats of the triplet of bases, CAG, whereas people with HD have a larger number of repeats of the CAG triplet. This is called a stutter. There is a rough inverse correlation between the number of times the triplet of bases is repeated and the age of onset of the condition.
Oh, you mean the more the stutters, the earlier the condition appears. Got it. So teacher, is Huntington's disease also expressed in homozygous recessive condition?
Not really. Huntington's disease is an example of a mutation that is inherited as a dominant allele. This means that most people with the condition are heterozygous and have a 1 in 2 chance of passing on the condition to a child.
Oh, wow. First, you don't know if you have a condition. In addition, you pass the condition as a dominant allele.
Want to learn more? We have additional learning resources to offer for Cambridge IGCSE and ASA level subjects. Register now for free.